Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video I am going to be starting chapter 16 of the AS and A-Level Biology syllabus, and that is inherited change. So in case you've been wondering why you look like your dad, why you have your mom's ears, why you have your dad's nose, this is the chapter where you learn about that. But it's also the chapter where you learn about gene mutations and how genes are controlled. So it's very, very interesting. And I find that students are typically amped for this chapter, so I hope that you will enjoy it. If you've just visited this channel, I think the key thing to point out to you is that it is a chronological order of all the AS and A-level biology content. But in addition to that, you can also use the playlists function to just check on videos. I have organized all the videos under the chapter title, so you can simply go there and look for the chapter that you might be interested in. Besides revision, this is also a good channel to expose yourself to content before you go to the classroom, so that you go to the classroom with questions rather than seeking to understand afresh. So I hope that you find those helpful. I'm not going to spend a lot of time with these introductory notes. Let us dig into inherited change. So the first thing that you would need to know as we start to explore this chapter is the concept of homologous chromosomes. So chromosomes, first of all, are structures within the nucleus that carry DNA, and they carry the genes that we express. And you would find as we go along that we have quite a number of chromosomes in the human cell, and they differ from what you would find in a dog, for example, or in a rat or in a bee, depending on what animal you're looking at. Homologous chromosomes are chromosomes that are very similar in structure. Not very similar, they are absolutely similar. So if you look here, for example, at the structure that has been put here on the slide, you will see that first of all, these chromosomes have the same central points right there. They seem to have the same length before the centromere, and they seem to have the same length after the centromere. Obviously, if you have watched the videos on mitosis, then this is not a strange thing to you. But just letting you know that homologous chromosomes are chromosomes that are very similar in structure and in composition. When we speak of a haploid number refers to the number of chromosomes in one set. So, for example, in the human cell, we typically say we have 46 chromosomes. 46 is what we refer to, I'm just going to use a white pen this time because this is a dark slide. 46 is what we refer to as a diploid number. A diploid number means that we have two sets of chromosomes, and don't worry, this becomes clearer as we go along. So if it's a bit confusing now, just hold on, I will explain it as we go along. So a diploid is two sets of chromosomes, and we typically refer to it as 2n, while haploid is usually just n, and it refers to one set of chromosomes. We also have a term called a zygote, and a zygote is a diploid cell that is formed from the fusion of two haploid gametes. So when we speak about things like gametes, we are referring to things like the ovum a woman would release and the sperm a man would release. When those two fuse together, they make a diploid zygote. But the interesting thing about them is that they are haploid, which means that it becomes n plus n, and that is what then gives us 2n. And so these are just key points for you to bear in mind before we go deep into this chapter. This here is what is called a karyogram. A karyogram simply is a photograph of all the chromosomes that are in a cell. If you look at this karyogram, something that is very peculiar that you notice immediately is that the chromosomes are arranged in pairs. And if you count the number of pairs that we have 23 pairs. This is 22, and then over here it says XY, that's the 23rd pair. Besides the fact that they are arranged in pairs, they also seem to be homologous, if you look at that. So basically, the homologous chromosomes are standing together in this karyogram. And what this is a karyogram of the chromosomes in the human cell. So we usually say we have 22 pairs of autosomes. So I'm just going to write that here. That's an A. 22 pairs of autosomes, and we have 46 chromosomes in total. So 22 pairs of autosomes and 1 pair of sex chromosomes. The sex chromosomes are the ones that determine your biological sex. So they determine if you're male or female sexually in terms of biology. while the autosomes carry other genes for other traits. For example, they might carry the gene for skin color, for eye color, hair color, all of those kinds of things. So the autosomes are 22 pairs, and obviously if you count 22 pairs, that would give you 44 chromosomes in total, plus one pair of sex chromosomes. That gives us a total of 46 chromosomes. Something that you also notice here is that the X chromosome is a lot bigger than the Y chromosome, and I will discuss more on that as I go on to the next slide. So here we go. The X and Y chromosomes are non-matching chromosomes, which means that they're very different from each other. The X chromosome is a lot larger. It's the big one here, while the Y chromosome is quite small. All the other chromosomes in the karyogram I just showed you are matching chromosomes, and again, they're called autosomes, while the X and Y chromosomes are called sex chromosomes. In females, you would usually have two X chromosomes. So a female's chromosomal denotion is XX, while for a male, it is XY, because males have one X and one Y chromosome. So chromosomes are usually distinguished by their banding patterns when they're stained, and that is how we're able to tell which ones are homologous and which ones are not. So this is just to show you how some chromosomes look, and this is an example of the X chromosome, for example, just showing you how the genes are arranged. So for example, here is a gene that enables the kidneys to retain phosphate. Here is a gene that controls the production of cytochrome B. Here is another gene over here. You can see that they're stained differently. This is the centromere rather, that's the centromere, but here's a gene here that controls the production of testosterone, and it goes on like that. This is how the genes are arranged on the typical X chromosome. And obviously, if there is a defect in the chromosome, that can result in certain kinds of diseases within the individual. For example, think about this over here. So over here, there's a gene that controls the production of factor VIII protein. And this is a protein that is necessary for blood clotting. If there is a disease or a disorder on this chromosome or on that section of the gene, then the person would develop hemophilia A. So the chromosomes play key roles in the sense that they carry genes that determine how we look, how we function, and how we go about our way in the world. Obviously, this is not some philosophical function that they're playing, but just to let you know that they're very important in determining whether or not a person has a medical condition or not. So before we get into all the fun stuff, I have to bother you first of all with meiosis. So in chapter 5, we started off with mitosis, or we discussed mitosis. And what we learned in mitosis is that a cell would divide and it would produce identical daughter cells that have the exact same chromosome number. And so for that reason, mitosis is for growth, it is for asexual reproduction. So if, for example, you fall down and you scrape your knee and your skin peels off, after a few days, your skin starts to grow back, and that is because due to mitosis, the cells are able to divide and ensure growth of the skin cells again. With meiosis, things are a little bit different. Meiosis is not a growth division. It is more of a reduction division. Meiosis is a nuclear division where you halve the chromosome number. So I'm going to use a red pen here, or rather yellow. I think yellow would be more visible. So you have the chromosome number when you do meiosis, which means that you start off with a diploid cell, and at the end of meiosis, you end up with haploid cells. Meiosis is mostly used during reproduction, but what we call sexual reproduction. So while mitosis might be for asexual reproduction, meiosis is for sexual reproduction. And as a result of that, the chromosome number has to be halved. The reason for this, and I often ask students this question, I say, hey, why do you think we have to halve the chromosome number? Well, now let's think about it this way in terms of the number of chromosomes. Okay, so a human cell has 46 chromosomes, just like we just learned. If this human cell undergoes meiosis, it will produce two daughter cells that have 23 chromosomes each. All right, so let's assume this is a female cell, and so in the ovum we have 23 chromosomes. If this ovum meets a sperm that has also undergone meiosis in the right way, it will also have 23 and 23 over there. And so this then means that the ovum would mix with the sperm, and that gives birth to a zygote that has 46 chromosomes. But now, if we do not, I'm just going to erase on the side over here. If we do not have the chromosome number, so let's say, for example, there's an error in the body, and so we end up with 46 chromosomes in the ovum, okay? And we then maybe mix it with a sperm that has had normal division of 23 chromosomes, we're going to end up with a chromosome number that is 69. Now, 69 chromosomes in an individual is an indication of a chromosome aberration according to biology, and that suggests that that person might become infertile because they're not considered to be the normal chromosome number. And so this is why it is important for meiosis to happen properly, so that the chromosome number can be halved and so that we can give birth to fertile, viable offspring as we go along. So again, this is just an explanation for that if a chromosome number is not halved, the number of chromosomes will double in every generation. And obviously that then becomes a problem, because look at this here. We start with N and N, that's 2 haploid gametes. They fuse to form a zygote. That zygote undergoes divisions, it grows and becomes an adult. And then now, if it has an error in meiosis and it makes only diploid gametes, then we're going to have a tetraploid, basically, which is an adult that has 4 different chromosome numbers, and that way we are unable to maintain the evolution. Well, I shouldn't call it the evolution, but we are unable to maintain the sustenance of the race, because obviously with 4N, I think tetraploids are usually fertile, but that can also then result in them matching with someone who's not a tetraploid, who's a diploid, and so that gives birth to a triploid. And I know that just sounds like gibberish to some of you now. Don't stress too much about it. As we go along, it would become a lot easier to grasp. So now let's look at the different stages of meiosis. The first thing I tell students when it comes to meiosis is to remember that meiosis occurs in 2 phases. You have meiosis 1 and you have meiosis 2. And as a result of that, you name the different stages of meiosis accordingly. So you say prophase 1 if you're talking about meiosis 1, and then prophase 2 if you're talking about meiosis 2. Another thing I tell students, and this is a good trick that might help you remember meiosis well, is that meiosis 2 is actually mitosis. So if you have learned mitosis, you will be able to remember meiosis 2. Meiosis 1 is also similar to mitosis. The difference is that in meiosis 1, you are dealing with whole chromosomes. There is no point in meiosis 1 where the chromosomes are separated into chromatids. Something else you need to take note of in meiosis 1 is the fact that you have crossing over that happens during meiosis 1, and that is an important thing in genetics. So now let's look at all the different phases. So you have prophase 1, and again, it's just what you've heard before, that the nuclear envelope disappears and you can have the homologous chromosomes that then appear and they pair up. All right, and in metaphase 1, you find that those chromosomes are more aligned and they have crossing over between their chromatids. What crossing over simply means, in very simple terms, is that the chromosomes exchange genetic information with each other. And so if you look here, you can see that the blue chromosome here is acquiring some purple over there, and the purple chromosome is also acquiring some blue over here. And that is metaphase 1, basically, and we call these bivalent chromosomes, and they would align or line up at the equator, which is again similar to mitosis. Then we go on to anaphase 1, where the whole chromosomes are pulled to opposite poles. So remember what I said at the beginning of the slide, that with meiosis, think of it as mitosis, but in meiosis 1, you're dealing with whole chromosomes, and in meiosis 2, you're dealing with it as though it is mitosis. And so you have the chromosomes are pulled apart, but again, take note, crossing over has happened, so you can see that the chromosomes have become a mixed bag. The reason why crossing over is so important is that it allows for genetic variation, and it allows us to have a mixture of alleles that then determine how we function or how we look. And this is very, very important for the survival of any species. And then we have telophase 1, where we have a nuclear envelope forming around the whole chromosomes. And at the end of that, we have cytokinesis, where we then produce 2 daughter cells of chromosomes. But these are no longer identical chromosomes as such. They are now chromosomes that have a mixed bag of traits because they've undergone crossing over. Once we finish meiosis 1, the cells then go on to meiosis 2. So now remember, we started off with 2 cells, all right? So 2 cells are coming in here. What's going to happen is that the first cell obviously will then go through prophase 2. Prophase 2 is like prophase of mitosis, which basically means that it's a nuclear envelope that disappears, the centrioles move to the opposite poles, spindle fibers start to form, same old things that you've always heard. And then at metaphase, the chromosomes will line at opposite poles. But if you look here, can you see here that the chromosomes have a mixed bag? So that red one has some blue on it, the blue one has some red, because they've had crossing over in metaphase 1. And then in anaphase, they are pulled apart by their kinetochores to opposite poles. So this means that we are basically doing mitosis in meiosis II. So always bear that in mind. And then we have, from there, we then have that cell splitting into two, and so we end up with 2 daughter cells from the first one and 2 daughter cells from the second cell. So remember, we came in with 2 cells from meiosis I, and now those 2 cells, each one has split rather into 2 different cells. And so at the end of meiosis, what we have is 4 daughter cells, and these 4 daughter cells are haploid in number. So they are not diploid, they are haploid, because the chromosome number has been halved. And the reason the chromosome number is halved is because when we get here into meiosis II, we have them being separated by sister chromatids, which means that we are splitting them apart and we are halving their number. And as a result of that, we end up with half the number in each of the different cells. And so we end up with 4 haploid cells at the end of meiosis. And again, this is just to show you what happens. So you can see there the difference between mitosis and meiosis. So in mitosis, you start off with a diploid cell of 46 and you end up with a diploid cell of 46. And the reason for this is that, remember that with mitosis, before anything happens, you have the G1 phase and then you have the S phase. These are the interphase subphases. If you haven't watched the mitosis video, I really recommend that you do so that this doesn't go over your head. But think about it, that you have the G1 phase in interphase where the cell grows, and then you have the S phase. And in the S phase, you have DNA replication, which means you double the number of DNA before you even do the division. And so when you do the division, you can have an extra copy of DNA to give to the new cell, which then keeps it as a diploid cell. In meiosis, there is no such thing. So we don't have that DNA replication happening before meiosis, which means that the cell simply undergoes all of these divisions. At the end of meiosis I, it will have 2 daughter cells that are diploid, and then at the end of that, it will have 4 daughter cells. That's at the end of meiosis II, 4 daughter cells that are haploid. So that is the difference between mitosis and meiosis. If you have any questions, please just post them in the comments, but I hope that that explanation was sort of helpful for you. So why is meiosis important? And I think I've sort of alluded to it already, that with meiosis, what we're trying to do is we're trying to form reproductive gametes. And for us to form these gametes and give rise to viable offspring or potential for viable offspring, we need to halve the number of chromosomes so that the next set of offspring will have the same number of chromosomes as their parents and not double the number. In order to achieve that, humans usually undergo something called gametogenesis. And just like the name suggests, gametogenesis is the formation of gametes. So in male, for example, it is referred to as spermatogenesis, if you're going to be specific, which means the formation of sperm. And for female gametes, it is called oogenesis, and that is just the formation of the ovum. So in sperm production, I'm going to show you an image that I got from the textbook that might just help you understand it better. But you have diploid cells in the testis that would divide to produce spermatogonia. These would grow to form primary spermatocytes, and then they undergo meiosis I and meiosis II, and meiosis II would then produce haploid spermatids that mature into spermatozoa. The fun fact about this, many people, I consider it a fun fact, but not many people would agree with me, is that for women, it's a little bit different. So in females produce all their eggs when they're born. So they're born with all the eggs that they would ever have. And so some of these eggs are not viable and they get lost as polar bodies, but some of them are viable and they get lost every month through menstruation. So I just thought to put that in here in case you didn't know that. And I'm sure you're just like, oh my goodness, if you're a female, you're probably horrified at that design. I also don't agree with it. Okay, let's end off here. This is just a schematic from the textbook. I really find this textbook very helpful because it has very descriptive images, and it also has like very easy language and details for students to follow. So this is spermatogenesis on the left over here. As you can see, you start off with a diploid cell that grows and then basically becomes spermatogonia, and then it becomes a primary spermatocyte and divides into secondary spermatocytes, which are haploid, and those secondary spermatocytes then give birth to spermatids that then grow to become spermatozoa. So that is what spermatogenesis in human cells would look like. Whereas with females, it's a little bit different. So you would have a diploid cell, and it's produced when the female is still an embryo, so when she's just been formed from the fusion of a male and a female gamete. And then you have all of these oogonia that result from that, and they grow and they become primary oocytes, and they are diploid at that point. But at that point, division also stops until the female reaches puberty. And once she reaches puberty, the primary oocytes would then divide again, and the half of that becomes a polar body that's lost, and the secondary oocyte that results, which is a viable egg or a viable possible egg, will then grow and then be released once a month as ovum. And if there's no pregnancy, then that is just lost in menstruation blood. So that is the end of this section of the chapter, and I hope you found it interesting, or at least it's piqued your interest in genetics. If you have any questions, or if I've made any errors, which I have realized I tend to do when I am speaking and not looking at students face to face, please point them out in the comments. It would be very helpful for others to be able to see where the mistakes are, and also for me to add corrective notes in the description. So if you notice an error in something, just look at the descriptive notes or the description of the video first. If you see that I haven't put a correction up, please draw my attention to it, and I promise to put it up as soon as possible. Thank you so much for watching, and the next video is a lot more fun because it is more about genetics and how we inherit genes. Until the next video, have a good time. Goodbye.